and today the title of my presentation is Anatomy of Flowering Plant. Yesterday, Kiran has covered the various concepts based on the morphology of the flowering plant that are all characterized with the stems, roots, flowers, fruits, etc. So by visualizing those components or an organ, we get the idea that so and so plants are fall under the category of angiosperm or a gymnosperm or any other classification norm. But what if someone gives you a permanent slide and tell you to identify the mounted tissue or a cell? What will you do? So for that purpose, we study the plant anatomy or a phytotomy. It's a general term for the study of internal structure of the plant, and it helps us to understand the difference in the structure and organization of the plant body and the specific function according to their modification and all. Right? So let's proceed further. This is all we are going to discuss now. That is plant tissue classification. So first of all, this tissue. These tissues are made up of a group of cells that should have a common origin and perform a common function. So this plant tissues are classified into two types, that is meristematic and permanent. This meristem word derived from the Greek word meristos, which means division, and this meristematic tissues are classified into three types: apical meristem, lateral meristem, and intercalary meristem. Whereas these permanent tissues are of two types: simple and complex. The simple tissue are again classified into three types: parenchymatous tissue, collenchyma, and sclerenchyma. Whereas these complex are of two types: xylem and phloem. These are the another components of this complex permanent tissue. So this is all about the tissue, that is a group of cells and common origin and usually perform a common function. So first we will discuss about the meristematic tissue. So this meristematic tissue shows the active division throughout the lifetime, and they are active cell. They have a specialized region in which the active cell divisions takes place. Right. So there are three types of meristematic tissues. This one, apical at the tip, intercalary in the middle, and the lateral at the sides. So the apical meristem, which are present at the tip of the root, are referred as root apical meristem, and the meristem which are present at the tip of the shoot are known as shoot apical meristem. These are the primary tissues. So this primary tissues, this apical meristem, are differentiated into primary meristem. Here we are talking about the meristematic tissue. So this is this apical meristem are differentiated into three kind of primary meristem, that is protoderm, procambium, and ground meristem. So this protoderm, first one is the protoderm that lies around the outside of the stem and develops into the epidermal layer of the plant. And this procambium, this lies just inside the protoderm and develops into the primary xylem, primary phloem, and it also produces the vascular cambium, cambium, and secondary meristem. You can see this image. Are you able to see this picture clearly? Primary meristem, protoderm, ground meristem, procambium. This ground meristem develops into the cortex and the centralized part of the plant, that is a pith. And they are composed of simple permanent tissues, which are parenchyma, collenchyma, and sclerenchyma. So we will discuss this later. There is an intermediate part during the shoot apical meristem, in which during the elongation of the leaves during the formation of the leaves, and elongation of the stem, some cells left behind and forms a shoot apical meristem that results in the formation of axillary buds. These such buds are present at the axils of the leaves, and that buds are capable of forming the new branches or a flowers or a leaf. So we can see this axillary buds are the formation of results due to the formation of due to the presence of dormant cells at the junction of the stem and the petioles, means those cells which remain dormant, that results in the formation of axillary buds. Now the second one is secondary meristem. This secondary meristem are of two types, that is vascular cambium and cock cambium. So these are also called as lateral meristem because see, they surround the established stem of the plant. Why they are referred as lateral meristem? Because they surround the established stem of the plant and they grow laterally, that is known as, which increases the girth of the plant. So that is why they are known as secondary meristem. So there are two types, vascular cambium and cock cambium. So this vascular cambium, which produces secondary xylem and secondary phloem, and this process that may continue throughout the life of the plant, and this vascular cambium is responsible for the formation of woody part in the plant, and such plants are known as herbaceous plants, and the plants which does not involves in the secondary growth formation, those plants are known as herbaceous plants. This does not occurs in the plant that do not go through the secondary growth. Those plants are known as herbaceous plants. Now the second one is cock cambium, which is also known as phallogen, which gives rise to peridome and replaces the epidermis just to provide a new protective cell layer. And we can see the image here. This cock cambium, the outer surface of the cock cambium is known as phloem, and the inner surface of the cock cambium is known as phalloderm, and cock cambium itself known as phallogen. So this phallogen, phloem and phalloderm collectively known as peridome, right? So if we are talking about the woody plant, so there is another concept of heartwood and sapwood. So this heartwood, you can see, are you able to see this darkish brown part? That is heartwood. Here also it is there, heartwood and sapwood. So this heartwood is a region comprises of dead element with highly lignified wall, and this heartwood does not conduct water, but it gives mechanical support to the stem. Whereas this sapwood, which is lightish yellowish in color, this region is responsible for the conduction of water and minerals from the root to the leaf. You can see this sapwood and heartwood. There is a difference in the color and the structure. This is a cock cambium, and these are the bark, which is responsible for the formation of the woody texture in the plants. So another one is intercalary meristem. This intercalary meristem, which occurs between the mature tissue, and this is responsible for the rapid growth. So they occurs in the grasses and regenerates the part removed by the grazing herbivores. And the third one is lateral meristem. The lateral meristem is a type of meristem that occurs at the lateral area of the plant, and thus it is responsible for the secondary growth of the plant, which means that increases the girth, diameter of the plant. And the lateral meristem is an example of secondary meristem. We have already discussed that secondary meristem is responsible for the formation of woody part, and that also helps in the increase in the girth size of the plant. So this lateral meristem is an example of secondary meristem. So till here it was all about the meristem and their three types. Now we will discuss about the permanent tissue. This permanent tissue, these tissues are completely grown up and have lost their ability to divide further. So they generally don't divide. They are now mature enough. So these permanent tissues are now classified into two types, that is simple tissue and complex tissue. The first, we will discuss about simple tissue. These simple tissues are made up of only one type of cells. It may be totally parenchyma or totally collenchyma or a sclerenchyma. So these simple permanent tissues are classified into three types. The first one is parenchyma. The cell of these tissues are living and with thin cell wall, and the cell can be oval or round or polygonal in shape, and it mainly act as a packaging tissue and provide mechanical support, whereas it also helps in the storage of food. But based on the specific function, when the parenchyma can be further divided into chlorenchyma and arenchyma. When these parenchymatous tissues are filled with chlorophyll, they perform the process of photosynthesis and they are referred as chlorenchymatous tissue. And when the parenchymatous cells are filled with arenchyma, they forms air channels and they are known as arenchyma. Now the second one is pollenchyma. These cells are living cells and have elongated shape, and the corners of the cells are thick, and you can see the corners of the cells are thick due to the deposition of hemicellulose, cellulose, and pectin. This pollenchymatous tissue can be oval, spherical, or polygonal in shape, and the function of this pollenchymatous tissue is to provide flexibility and mechanical support to the plant. And this pollenchymatous tissue can be located in the leaf stalk and below the epidermis. This is the image of the pollenchymatous tissue. And the third one is sclerenchyma. The cell of the sclerenchymatous tissues are dead. The cell wall is very thick due to the deposition of lignin, and they have few or numerous pits. You can see few to numerous pits, and they are generally located in the leaf veins and hard covering of the seeds, and can also be found surrounding the vascular bundles. The main function of the sclerenchymatous tissue is to provide rigidity and mechanical support to the plants. This sclerenchymatous tissue are of two types, fiber and scleroid. These fibers are thick-walled, elongated and pointed cells, generally occurring in group and in various parts of the plants, whereas these scleroids are spherical, oval or cylindrical and have highly thickened, and they are dead cells with a very narrow lumen cavity. You can see the lumen is very narrow as compared to fiber, and they are commonly found in the fruit wall of nuts and pulp of fruits like guava and pear. Now this was all about the simple permanent tissue. Now we will discuss the complex permanent tissue. These complex permanent tissues are made up of more than one type of tissues, and these work together as a unit, and they coordinate with each other to perform specialized function. You can see the specialized function in the plant body, which are constituted by the two types, xylem and the phloem. So the xylem tissues is responsible for the conduction of water and minerals from the root to the leaves and the stem, and it also provides support to the plant. And it has four elements, that is, there are tracheids, vessels, xylem parenchyma, and xylem fibers. So these tracheids are elongated. You can see they are elongated or tube-like cells with thick and lignified walls and with a tapering end. And these are dead. And the vessels, when we talk about the vessels is long cylindrical tube-like structure made up of many cells. And the xylem fibers are highly thickened cell wall, and this xylem parenchyma cells are living and thin walled, and their cell walls are made up of cellulose, and they store food materials in the form of starch or else in the form of fat and other substances like tannins. So this is the image of the xylem with their vessels. And this primary xylem, when the xylem forms, the primary xylem, they forms in two types, protoxylem and metaxylem. So in stem, this protoxylem lies towards the center, whereas the metaxylem lies towards the periphery. So this type of primary xylem is known as endarch, and in root, the protoxylem lies towards the periphery and the metaxylem lies towards the center, and they are referred as exarch. Now, this was all about the xylem. phloem, this complex permanent tissue helps in the translocation of food, that is.
कहाँ से sound आ रहा है? Background sound?
सर वो फोन आया.
अच्छा.
This complex permanent tissue helps in the translocation of food that is prepared by the photosynthesis in the leaves to the various parts of the plant. And this phloem consists of 4 elements that are sieve tubes, companion cells, fluent fibers, and fluent parenchyma. So you can see the sieve tubes. The sieve tubes elements are also long tube-like structure arranged longitudinally and are associated with the companion cells. You can see now the companion cells are specialized parenchymatous cells which are closely associated with sieve tubes elements, and these sieve tube elements are connected with a common longitudinal wall. This sieve tube and a companion cell shares a common longitudinal wall, and whereas this fluent parenchyma, this is made up of elongated tapering cylindrical cells which have dense cytoplasm and nucleus, and the cell wall is composed of this cellulose, and the pits is present in between, which is responsible for the plasmodesmatal connection between the two cells. And the last one is fluent fiber. This fluent fiber is also known as bast fiber, and they are made up of sclerenchymatous tissue, and they are generally absent in the primary fluent. Means the presence of fluent fiber or a bast fiber is the characteristic feature of secondary fluent. The presence, you can see this fluent fiber is present only in the secondary fluent. Now, till here it was all about the tissues and their functions. Now we will discuss about how these tissues work together and forms a system. So there are 3 types of plant tissue: epidermal tissue, ground tissue, and vascular tissue. Now the first one is epidermal tissue. They forms the outer covering of the plant body, and they are comprises of epidermal cells, stomata, and epidermal appendages like trichomes and hairs. So these trichomes are responsible for the production, and it also helps in retaining of water. Excess loss of water are prevented by the presence of trichome, and these epidermal cells are parenchymatous with small amount of cytoplasm lining the cell wall and a large vacuole. This epidermis of the plant leaves secretes a coating called cuticle that helps the plant retain water, and the epidermis in a plant leaves and the stem also contain a minute pores that are known as stomata. So these stomata are very minute pores present in the abundant amount at the position of the leaf, whereas it is all very few in case of stem and almost negligible in case of root. And this you can see this stomata are made up of two minute kidney shaped cells known as guard cells, and these guard cells are surrounded by the accessory epidermal cells known as subsidiary cells. These guard cells, you can see, these guard cells are living cells, and they contain chloroplast. So they are responsible for the process of photosynthesis, and they also help in the gaseous exchange process. Here, the image of the guard cells, which shows the inner wall of the guard cells is thicker, and the outer wall of the guard cells is comparatively thin. So this is to maintain the turgidity of the stomata during the water absorption. And here there is a difference in the shape of stomata in dicot and monocot. In case of dicot, the shape of the guard cells is kidney shape, whereas in monocot it is dumbbell shape. So these guard cells in the epidermis regulate the gas exchange between the plant and the environment by controlling the size of the stomatal opening and closing. Now the ground tissue. We have already discussed about these simple tissues. The ground tissues are made up of lot of simple tissues. These tissues are composed mainly parenchymatous cells and some amount of pollen chyma and sclerenchymatous cells. So this parenchyma cells synthesize and store organic product in plants, and most of the plant metabolism takes place in the cells. We have already discussed, so that's why I didn't included image again here. So we will just read this. So this parenchymatous cell in leaves control photosynthesis. This collenchyma cells have a support function in plants, and particularly young plants, just because this collenchyma cells gives flexibility to the plant. Now the third one is Sclerenchyma cells. It also have a support function in plants, but unlike collenchyma, they have a hardening agent and are more rigid cells. Now the third type of system is vascular system. This vascular system, vascular tissue, is composed of xylem and phloem. This tube-like structure transport water and nutrients throughout the plants. And this xylem, they allow the water and the other nutrients to be transported throughout the plants. And the xylem consists of two types of cells known as tracheids and the vessels element. Vessel elements include xylem fiber and xylem parenchyma. The tracheids and the vessel elements form the tube-shaped structure that provide pathway for water and mineral to travel from the root to the leaves. While these tracheids are found in all vascular plants, and vessels are only found in the angiospermic plants. So the presence of water is the characteristic feature of angiospermic plants, because the vessels are absent in gymnospermic plant. Now phloem. Phloem is composed mostly of cells called sieve tube cells and companion cells. These cells assist in the transport of sugar and nutrient produced during the photosynthesis from the leaves to the other part of the plant, while the tracheids cells are non-living and sieve tubes and companion cells of the phloem are living. So these companion cells possess a nucleus and active transport sugar into and out of the sieve tubes. So here it was all about the tissues and their system, how they are modified, what are their characteristics, and where they locate, and how they function. Now, according to these tissues and system will remain present in all of the plants, and as we know that angiospermic plants are of two types, dicots and monocots. So now we will discuss about the dicots and the monocots plants. So first we will see some basic parameters of comparison between dicots and monocots. So we will start with the definition. The plant with seeds having only one cotyledons are called monocot, and the plant seeds having two cotyledons are called dicots, as the name suggests. Right? So these cotyledons are the part of the embryo within the seeds that act as an initial energy source for the plants. And this cotyledons of the monocot seeds is rather thin and does not contain sufficient food material, while in the case of dicots plants, dicot seed, they are fleshy and they contain enough nutrient. You can see the monocot and dicot seed. Now the second difference is in the leaves. The veins of the leaf of monocot seeds plant are parallel, and you can see the parallel venations are present in the monocot plant, and the veins of the leaf of dicot seed plant have a net-like structure. This messy net-like structure of venation is present in case of dicot leaves. Now the third difference is in flower. The flowers are present in the multiple of 3. Here, multiple of 3, and the flowers are present in the multiple of 4 or 5 in case of dicot. Now the third difference is in stem. The vascular bundles are scattered. You can see the red dot represents the vascular bundle, which is scattered in case of monocot, and the vascular bundles are present in a ring-like pattern, which in case of dicot. And the roots, the fibrous roots are present in the monocot, and the tap root-like structure are present in case of dicot. So the corn, wheat, rice, sugarcane, banana tree are the examples of monocot, and the mango, orange, tomato, beans, and pea are the example of dicot. So According to these are the external differences or morphological differences between the dicot and the monocot plants. Now we will discuss about the anatomical difference between dicots and the monocot. So first one is the anatomy of dicot root. So the outermost layer is made up of epidermis, the innermost layer is endodermis, and that are comprises of single layer of barrel-shaped cells, and it has waxy material known as suberin in the form of Casparin strips, and next to the endodermis lies a few layer of parenchymatous cell, thick few layer of parenchymatous cells which are known as pericycle, and here the pith is small in case of dicot, whereas in case of monocot you can see the pith is large and usually have more xylem bundles as compared to dicots. We can see they have lower number of xylem bundle, whereas in case of monocot they have more xylem bundles as compared to dicots, and the monocotyledonous root do not undergo any secondary growth. Here they undergo the secondary growth, and this is the general difference between the dicot root and the monocot root. All of the parameters remain same. In case of anatomy of monocot root, it is similar in case to the dicot root in many respects, like it has epidermis, it has cortex, endodermis, pericycle, vascular bundles, and pith, the same which is present in the dicot, but it has little difference between the monocot and the dicot root that we have discussed, that pith is large and usually xylem bundles are present in more amount. That's all. Now we will discuss about anatomy of monocot stem. See, this dot represents the vascular bundle. This, in case of monocot stem, these vascular bundles are scattered. These are scattered and they are conjoined and closed, and they are surrounded by the sclerenchyma tissue. Whereas in case of dicot, the vascular bundles are arranged in a ring-like fashion. So the ring-like arrangement is the characteristic feature of the dicot stem, and the vascular bundles is conjoined and open. This is the basic difference between dicot and monocot stem when you take a cross section. Now we will discuss about the dorsiventral section of the dicot leaf. So the basic difference between the dicot leaf and monocot leaf we will discuss right now. See, this bundle sheet of the dicot plant's leaf generally have a single layer and form a colorless cells. This bundle sheet cell surrounds this vascular bundle which contains xylem and the phloem, whereas in case of monocot, the vascular bundles single or double layer and form a colored cell due to the presence of chloroplast. So the vertical section, this is the vertical section of dorsiventral leaf, and it contains three parts, namely epidermis, mesophyll, and the vascular system. So this epidermis, which covers both the upper surface, that is adaxial epidermis. See, you can see adaxial epidermis, and the lower surface is abaxial epidermis. Abaxial epidermis. Abaxial epidermis. So this abaxial epidermis generally bears more stomata than the adaxial epidermis, and the tissue between the upper and the lower epidermis is known as this mesophyll cells. So this mesophyll cells possess a chloroplast and carry out photosynthesis, and they are made up of parenchymatous tissue, and it has two type of parenchymatous tissue, that is palisade parenchyma, which is made up of which contains mesophyll cells, or else spongy parenchyma, or else spongy mesophyll cells. So this adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically. You can see which are arranged vertically and in a parallel fashion. They are elongated cells and they are arranged vertically, and they are all parallel to each other. And the spongy, when we talk about the spongy cells, they are oval round and loosely arranged cells, and they possess air cavity in between, and this air cavity provides a large space, and this vascular bundle are surrounded by a layer of thick, we have already discussed this thick layer of bundle sheath cells. So this was all about the dicot leaf. Now now we will discuss about the transverse sections of monocot leaf. So this mesophyll are not differentiated in palisade and spongy cells in case of monocots, right? And certain adaxial epidermis along with veins modify themselves into large empty colorless cells. These cells are known as bulliform cells. You can see the empty space that are these are the region of bulliform cells, and these bulliform cells are responsible for the maintenance of water level in the plant. So when the plant absorb water, and so the leaf become turgid and the leaf surface are exposed, and when they are flaccid, it means there is a lack of water or the plant is in a water stress condition. So then they make the leaf curl inward to minimize the water loss. So this was all about the function of bulliform cells and the transverse section of monocots and the difference between dicot leaf and a monocot leaf. Now here is the question. Test yourself.